All right, I'd like to introduce my friend and colleague, Raphael Santos. Raphael is a visiting scientist here at CDU. He's an aquatic biologist, an ecotoxicologist, and interested in conservation biology. Like a lot of people who come to Darwin, Raphael came to Darwin only traveling through and ended up staying here for almost over a year now. And is almost a local Darwinian. And we both work on similar topics. So Raphael and I have been talking about all things fish and water. Today he's going to give us a presentation about trends in European freshwater fish populations and the implementation of ecological risk assessment methodologies in monitoring programs. So without further ado,
Thank you, Keller. Thank you for inviting me, because it's a great opportunity for me to have the chance to give you a seminar. It's been a while since I had such an opportunity, more than 2 years and a half. So thank you for inviting me. To start, I would like to give more details about who I am, my profile, and what I'm doing here in Darwin, because as Keller said, I'm traveling. So as we cannot travel at the moment, it's a good opportunity for me to make you travel to Europe. Oh yeah. So a long time ago, I started a master degree in marine biology in Brittany, and a second master degree at the École pratique des hautes études in Paris. So so here are just some picture of Brittany, which is a very beautiful place to visit if you go there, if you go to France. And after my master's degrees I started my PhD in aquatic ecotoxicology in 2 research centers, one in Paris called INERIS, and the other one is ELENA at the University of Lyon. So here are a picture of Lyon, and which is also a nice place to go if you travel to France. And after my PhD in ecotoxicology, I decided to work in closer collaborations with environmental managers, and I designed and I led and supervised several projects during my postdocs. And there I done this project at the Center for Functional and Evolutionary Ecology in Montpellier, and at the University of Applied Sciences, Western Switzerland in Geneva. So here are a picture of the Geneva Lake with a beautiful view from Lausanne, and here are pictures from the south of France. So after I finished my project in 2019, I wrote the final reports, and so I had just to write my papers because we had a lot of data to publish. And to do that, I decided to also travel to Melbourne, to Australia, to visit my colleague, Kirsten Paris, and to eventually start collaboration together. So I arrived January last year, and when I arrived to Melbourne, there was bushfires, and then a global pandemic started. So I was thinking, oh, what's going next? Maybe a few months, I will be stuck for a few months in Melbourne in lockdown. So as I couldn't go back home, I decided to travel to a nice place to live there and to write my articles. So that's why I'm in Darwin. So now you know everything about me. So let's talk about science. So we are going to talk about aquatic ecosystems. So as we know, aquatic ecosystems and especially rivers are under multiple pressures from human use to for agriculture, water consumption, from overexploitation or water pollution. So these ecosystems are under several pressures. And if we focus on freshwater fish species, in Europe, they are considered as the second highest threatened taxa, with 37% of these species that are threatened to extinction, classified as vulnerable, endangered, or critically endangered by the IUCN. When I give a talk in Geneva, I like to show that among the 14 species that are already extinct from the environment in Europe, these species, Coregonus fera and Coregonus yemalis, represented 70% of the fish catches in the Geneva Lake in the 20th century. A few decades after that, they were declared extinct. So it's just to make it a bit more real that the decline of biodiversity is also something around us. So if we think about the IUCN Red List status, these statuses are based on several factors that could be from the geographic range, could be based on the population size and structure, population decline, population dynamics criteria, or ecological traits and other factors. And in Europe and in US, small size, short life cycles, freshwater fish species are considered at higher risk of extinction due to their small geographic distribution range and their high level of endemism. But a small distribution area doesn't mean that the species is declining. So for sure, the species could be at greater threat of extinction because of small distribution area, but there might be not declining. So, and what about common species that are not threatened to extinction? Because by definition, they are common species, but they could be declining nonetheless. It's even important to think about population dynamics, as we don't have a lot of information about population trends in Europe, and even at the larger scale globally, we don't have I mean, maybe in Australia, we don't have much information about freshwater fish population trends. So based on this statement, I decided to work during my postdoc with environmental manager from the French Office for Biodiversity, and we worked on their data sets so luckily since 1990 they ran a standardized they ran a monitoring program so every year or every 2 years they sample fish in river through electrofishing standardized protocols, and I had access to their huge data set of 546 sites spread around France. And I focused my work on common species, as I told you. I focused my work on most common species, the 18 species that represent 94% of fish species and 88% of fish biomass that was sampled in this monitoring program. So by definition, they are common. And amongst these 18 species, only Anguilla, the European eel, benefits from a IUCN Red List status of critically endangered. But the other species are considered at least concern for now. But it's important to assess trends also in common species, because even slow variation in their abundance could lead to substantial modification of the aquatic ecosystems, because basically they shape, these common species shape fish communities. In this work, we had access to several data sets. The first, that we had access to data about the 18 species, about their life history traits and about their ecological traits. And we had access about, and so I used the data about fish abundance, about several parameters: fish abundance, fish biomass, and fish body length. It's always interesting when we analyze data sets to try to analyze several parameters to reach conclusion, especially if so it's even better when the three indexes or the three set of data give the same information. So to analyze this data set, we use state space model that are very interesting to widely used in population dynamics. This model make it possible to take in consideration an observation error, because there is because if you are aware about electrofishing programs, and there could be a high diversity, high variability due to the in-catch, due to stochasticity, or so we took in consideration this sampling error. But the most important parameter I want you to keep in mind is the population, is the growth rate. The growth rate if this R value is positive, it means that the population is increasing in abundance or in biomass, or if, like in this example, and if the population growth rate is negative, it means that the species is declining. To analyze fish length, we use Cantel regressions, which is an interesting tool because modeling trends in mean value is not always the most informative parameters, as we can see here. So on the left side, so here are data from all own data sets. As we can see here, if we model the trends in mean values, in median values, or any of these quantile, we would have the same information. But about this species, which is probably Scardinus erythrophthalmus, we can see that the information is different if we focus on the trend of the biggest individuals or in the trend of the smallest individuals. And as we had at the end a lot of results, including the 18 species, to take a step back on the results and investigate correlation with ecological traits, I computed principal component analysis. So here are the results in orange. So these are the posterior distribution of the population growth rate at the national scale, which means that if this value is below 0, it means that the species is declining in abundance in orange or in biomass in green. If this value overlaps 0, it means that this population is stable, as this species is stable as a national scale. And if this value is above 0, it means that this population is increasing in abundance or in biomass. To summarize these results, here is a representation with a picture of this fish. So we demonstrated that since 1990 in France, 11 species decline in abundance, 7 are stable or increase, and 14 species decline in biomass, and only 4 of them were stable or increase in biomass. So the next step, now we made a statement about the trends, but now it's the next step is to understand what are the ecological traits that make some species more vulnerable. Why do we observe such a variability? Even if most of these species are declining, some of them are still increasing in abundance and biomass. So that's why we focus on ecological traits. And as you can see on the result of this principal component analysis, biomass growth rate and growth rate in abundance are correlated, highly correlated, but they are also inversely correlated to fish life history traits and fish length trend, which means that we interpret that as that small fish species with fast life history strategy were stable or increasing in abundance and biomass, while larger fish species with a slow life history strategy were declining in France. It's confirmed by the basic linear regression we implemented using the population growth rate at the national scale. Here are the results. So the fast life history strategy species had higher population growth rate at the national scale, while slow life history strategists were more declining. And it's even clearer in biomass than it is in abundance. And it's also correlated to the size of fish. Like small fish exhibited more stable population, while bigger fish were declining. So in frequently disturbed areas, slow life history strategies are predisposed to population decline because it's due to their low intrinsic maximum population growth rates that are limiting their ability to recover after perturbation. Should I say that fast life history strategy are small fish species that reproduce quick, and have a short life duration. Like this species if we take the example of Cochus gobio, the lifespan is for 3 or 4 years, and they reproduce in the first year. While the species some of these species around here live around 15 or 10, 12, 15 years and reproduce after 4 or 5 years. So after perturbation, even if we mitigate the pressure, it takes time to recover. So so that's the work we've done at the national scale. But when we talked with the environmental managers, they were more interested to know what are the population trends at a lower geographic scale. Because in Europe, within the European Water Framework Directive, the management unit is the river basin district, and 200 river basin districts have been designed around across Europe. And so we focused on the river basin district in France, and we investigated population trends in the 5 main river basin districts. We excluded this one because of we had not enough data. And we had very interesting results because for some species like Anguilla, we demonstrated that the species is declining in abundance in the 5 river basin districts. And it wasn't a surprise because it's well known that this species is declining, and it is classified as critically endangered, currently under European regulation to restore European eel population. And we know that this species accumulate risk factors of population decline. I don't know if you are aware about the life cycle of European eel, but it's really fascinating. This species reproduce in Sargassum Sea in Caribbean, and then now they migrate to Europe around the Gulf Stream to colonize European river. They start before they reach freshwater, they metamorphosis and to be able to live in freshwater, and they live for 9-20 years in European rivers to grow. And after, usually females start their migration to go back to the Sargassum Sea after between 14 and 20 years. And and so after this long time in Europe, they start their migration to go back to the Sargassum Sea to reproduce. But the journey to go back to Caribbean is kind of dangerous because all along the way, the adults could be killed from many threats, like they have to go through dams, and so the adult mortality is kind of high. And the particularity is that when they start the migration, they stop feeding, so they rely only on the energetic reserve they accumulated during their lifespan in Europe, which means that if they accumulated reserved lipids in contaminated area, lipophilic micropollutants could bioaccumulate. And when they start their journey back to the Sargassum Sea, as they use their lipidic reserves, these micropollutants could be released in the body, and so so they will have to deal with these micropollutants on the way back. It was one of there are some interesting hypotheses that it could be one of the main threats of European eel. So this species basically accumulates risk factor of population decline. But for other species, we found about the European eel, it was not surprising, but about other species, we highlighted that there was a spatial diversity in terms of population trends. Like if we have a look on Scardinus erythrophthalmus, this species is declining in the Loire-Bretagne, but not in Rhone-Méditerranée. And one of the most interesting results was about Cochus spp, Cochus gobio. This species is the only one that is increasing in abundance and biomass at the national scale. And but in the Adour-Garonne river basin district, this species is declining. And it's interesting because it's mentioned Cochus spp as a taxa, because when they started the monitoring program in 1990s, they consider Cochus gobio as only one species distributed in France. But since we improved genetic analysis, researchers discovered that there is 5 or 6 species of Cotus in France, distributed in France. And Cotus aturi, that is considered as nearly threatened, is only distributed in this area, which means that as it is the only area, the sole area where river basin district where the species is declining, it would be interesting for environmental manager to extract data sets, focus on this species, on this river basin district, to check if Cotus aturi should be classified not as nearly threatened, but are threatened to extinction, and also to mitigate the pressures. So we made this statement about population trends at different scale, but so now the next step is to understand what are the pressures that are the most impacting fish population dynamics, fish population in Europe or in France. Similar pressure are impacting fish population in Europe. And it would be also relevant to investigate what are the demographic components that are the most impacted. So we could mitigate the pressure and probably design conservation program differently. But so that would be an option. That's something we should do, and I'm working on that. But what would be interesting is to find tools to prevent the degradation of fish populations before they are impacted. So as we know that preventing is always better than curing. And that's the second part of my research profile that is focused on river contamination and ecological risk assessment approaches. If you have any questions on the first part, maybe we could talk about that for a few minutes and switch from ecotoxicology after that. Do you have any question? Or would you like to, I keep going and...
I have a question. I was just wondering with the increases and decreases in population growth rate of the different species of fish, if you had a chance to look at like one of the slides that you presented, you had the increase in Cotus gobio in certain catchments, but not others. I was wondering if that correlates at all with declines in other species. So like large predator species, you had esox and those sort of species there. Could some of those relationships be explaining potential patterns as well?
Actually, I was expecting that there was it would be interesting to investigate this dynamic of in fish communities. But recently, I mapped using all this data, and there are areas where most of the species are declining. All of them, predator, prey, and that would be. So I didn't expect that, but it might be that we observed in some area in France a general decline, and it's not linked to dynamics a bit in fish communities, but it's definitely the next step. To investigate more on the different sites, what was the dynamics of. I forgot to put this map, but we could spot on this map that there are area where most of the species are declining, and it's kind of clear now. The next step is definitely to find why and to understand what are the pressures that make all of them declining, and could be contamination, but could be hydromorphological pressures or. Yeah.
Thanks. Yeah, really interesting.
Yeah, Veronica.
Other questions?
Thank you, Raphael. Yeah, this is very amazing.
It's not over.
I know, but this part is very amazing, and I'm gonna query something on that. Do you know anything about ecopath? Because I think that you sort of data that what I can see, it could be useful on doing a whole trophic analysis. Okay. Because if you have biomass and abundance of all these 18 species, Ecopath is a model that allows you to analyze the interaction, the trophic interaction between the different groups.
Okay.
And you can also add the human components such as fishery, as that is taking both the abundance and the biomass. So that could be a very good add-on to your project.
Okay.
And that is actually based in Europe. So most of the people working on Ecopath are based in Denmark. And it will be a very good way to understand whether some of the changes that are happening within the populations are happening because of intrinsic changes within the population, or is it because of human or changes in the ecosystem that they are working on. So Ecopath at the moment has an eco simulator and an eco space. So all of those things could be put together in which you can just check all these 18 species and see how the interaction, because once if the smaller animals are staying stable in the population and the bigger ones are the ones that could be a human impacted part because of fisheries probably, and you can put all of that into the system to try to analyze how all of this is interacting with the environment on the surroundings as well.
And also data about invertebrate, macroinvertebrates, and so it would be interesting to use these different compartments. And okay,
it goes to all the level. So it goes from it works on trophic groups more than on the species level. So you have to put them all together at the omnivores, the herbivores, the piscivores, and all of that. And of course, you have to put into the whole microinvertebrate as well. And then you get an amazing understanding of a box understanding of how the system is working.
Okay. Thank you, Veronica. We talk more about that later. Thanks. So yeah, we switched to a topic that is more focused on ecotoxicology. And because even if it's on ecotoxicology, my work was still focused on environmental, in collaboration with environmental managers, and also to improve policy, and also in the context of the European environmental policy. So as you know that more than 100,000 chemicals are referenced in Europe, but from 37 to 77 of them are daily used. And they unfortunately end up in rivers through direct discharge in water stream, through atmospheric deposition, or through water runoff of contaminated water. In Europe, we assess the chemical status of rivers and also the ecological status. It's the two main tools for environmental managers to detect disturbances. So the chemical status is based on the monitoring of 56 priority hazardous substances, and the ecological status is mostly based on the assessment of several ecological indicators such as diatoms, macroinvertebrate, or fish indexes. But it's important to think about the limitations of these tools. If we think about the chemical status, there is only a limited number of micropollutant that is monitored, only 56. And it started with 33, but we can't infinitely increase the number of substances that are monitored because of the cost of monitoring programs and because of technical challenges. And about limitation of the ecological status, we know if we focus on fish, we know that the fish-based index lack of sensitivity. It responds well to strong perturbation, to detect strong perturbation, but there is a lack of sensitivity when fish communities are slightly degraded. So that's why during my PhD, we focus on biomarkers and we propose to use biomarkers as complementary tool to ecological and chemical analysis. I don't know if you are aware about biomarker, but there are biochemical, cellular, physiological, or behavioral variation that can be measured in tissue or body fluid samples, or at the level of the whole organism that provide evidence of exposure to and/or effect of one or more chemicals. To make it simple, biomarkers are biological parameters that could be measured in fish. We could sample, for example, fish and investigate the induction of enzymatic activities in liver, enzymatic activities that are involved in contaminant metabolization and oxidative stress. We could eventually use a sample muscle to investigate enzymatic activity inhibition linked with neurotransmission failure, such as that could be linked to exposure to some pesticides. We could eventually investigate the level of DNA damage in erythrocytes due to genotoxicant pressure, or use histological analysis to investigate endocrine disruption. So there are plenty of biomarkers that could be used, from the omics tool to histological analysis. But the most important is to investigate, is to use several biomarkers in a battery in combination to assess the whole health status of organism. We propose this complementary approach to combine together several lines of evidence, because in ecological risk assessments, just like what I did with investigating biomass abundance and fish length, is try to get information from several lines of evidence to make conclusions. And it's the same principle here. Using chemical indexes, we could design chemical indexes, fish-based indexes that already exist, and biomarkers index to incorporate this analysis in a decision matrix that will make the decision making easier for environmental managers. So based on this statement, we implemented some field work during my PhD. So I sampled fish, three spines stickleback in French rivers in 2 reference sites, Petit Terrain and Oudancourt, and 3 contaminated sites that are under agricultural or urban pressures. And we investigated 9 biomarkers responses that are involved in pollutant metabolization, oxidative stress, neurotoxicity, and genotoxicity. And in this work, we highlighted that there was a correlation between the level of DNA damage and the T-bars and the erode parameters, which mean that fish have been exposed to contaminants that induce genotoxicity and oxidative stress. And we mostly discriminated fish on a urban pressure gradient. So these fish suffer from physiological disturbances, and this one as well. We use this data as 4 of these sites were at the moment part of the national monitoring program. We had access to chemical analysis and of the fish-based index. And when we gather this data on a decision matrix, it's something kind of simple. We realized that it was relevant for environmental managers to make decisions. So in sites like in Réveillon, when all the indexes show that there is a perturbation and disturbance, this site is in the suburb of Paris. Maybe it's not the most relevant one to focus to invest time, money, and to manage and to try to restore the area because it's already highly degraded. But if we apply the policy that preventing is always better than curing, in this site of Ronel and Scarpe, we demonstrated that even if the chemical index didn't show that there was contaminants in concentration at risk for aquatic organisms, fish physiology reacted through the several biomarkers we investigated. And it was interesting because the year we investigated we carried on this study, the fish-based index that was in a good status before our analysis turned to be in a mediocre status. So it means that biomarkers responses acted as early warning signals that point out biological disturbances in the individual scale, and so it could be useful to detect there are early warning signals before the community are severely affected. So this approach could be used in several contexts, using biomarkers to detect physiological disturbances. And some of the projects I led for the last few years were focused on the impact of sediment resuspension due to hydropower production. So we worked with hydropower companies, EDF and CNR, that are the 2 main companies in France, to investigate what are the threshold about sediment concentration that could have an impact. Because for now they produced hydropower thinking on thinking more about productivity, but now their activities, they try to make it more eco-friendly because of the social pressure and because environment is getting more important, and because of they are aware of the biodiversity decline. So based on a previous publication from Switzerland, we optimized his apparatus to resuspend sediment, and we ran an exposure for 28 days and 14 days in recovery. And so it's a very interesting tool to expose fish simultaneously to 12 tanks, but we can make it bigger. And so as you can see here, but if we check the to 2000 milligram per liter. It's so I wrote the paper about the results. We investigated at several we investigated biomarkers in like gill morphological alteration, oxidative stress, and genotoxicity in these fish. And but the goal is to improve the model that is currently used by environmental managers to manage sediment dredging. This model have been implemented, have been calculated in 1996, and it doesn't take very well in consideration sublethal effects. So it's time to update this model to run their activity, to define the threshold more properly. And to do that, we definitely need to do international collaborations. We designed the apparatus. We ran an experiment, like it's a good basement to start international projects, especially as this apparatus could be duplicated everywhere in the world anywhere in the world to investigate several concentration, the effect of sediments on several species. And what do we need is we need data to be able in 10 or 12 or 15 years to update this model. But we need data, definitely. And that's why this project was interesting. That's a good start for international projects. And that's the last project I would like to show you. And it's one of my, I really like this project. I didn't do it myself, but I supervised Abi Houdi, who is a Tunisian student. And she wanted to study ecotoxicology and not focus on fish, but on bird species. So we designed a project on common terms. She was studying at the University of Gabes and living in Tunisia. So here are places where there are several colonies of tern. One in Monastir that is a reference site, a nesting colony in a contaminated area, and in Chat Salem, which is the highly contaminated area. Gabes at this time and still is one of the most contaminated area in the Mediterranean basin. So what she did is that she monitored she checked when chicks were hatching, and after 3 weeks, she sampled blood to investigate several biomarkers of oxidative stress, but also there is one biomarker of genotoxicity. And we did almost the same thing that I did with fish. That we use this data to try to discriminate fish chicks from with a bad health status from chicks that suffer from physiological disturbances. And here we demonstrated that there is a correlation between antioxidant defenses and inhibition of that is inversely correlated to biological damages. And that is a marker of oxidative stress, and that's a marker of genotoxicity. And it's all and it's linked to the level of contamination in the area. So this work was very interesting, especially as I use this first dimension of the principal component analysis. I extract the data and make just a simple linear regression to investigate if there was a correlation between chick physiological status and their mass after 3 weeks. And actually, there is a significant correlation between the physiological disturbances physiological status and mass. So the chicks in a good health status gain more weight after 3 weeks than chicks that were exposed to oxidative stress and genotoxicity. And during this, it's very interesting because when we work in biology, it's very hard to extrapolate our results from several biological level. Like molecular biologists couldn't say that it's hard to say if the gene expression they measure could be linked to long-term population defects. And it's a challenge for ecotoxicologists to make the link between biomarkers and information at higher level. And we know that in birds, the mass of chick during early life stages is correlated to their long-term survival. So that's the first step to try to make the link between biomarkers and higher biological and disturbances at higher level. And that's just to finish. It's during a meeting. Abir said that when we started the project, Abir said that the chicks on the contaminated sites look very ugly. So yeah, look very ugly. But what do you mean, Abir? What? And yeah, compared to the chicks in the reference sites, these chicks are very cute, and actually they are. But yeah, she couldn't tell me what ugly means for her. And at this time because it's hard to explain, but at this time I watched a documentary about human beauty, and the conclusion that was the perception of human beauty is also linked to the symmetry of our face. And based on that, we check on the literature, and some researcher used asymmetric index of asymmetry to characterize morphological abnormalities. And so what she did is that when she made the blood sample, she also measured different parts of the body of these chicks. So here are the level of asymmetry. If it's around 0, it means that these chicks are symmetric, and I mean the left side and the right side. And so in the monastir, that is a reference site, the variability is quite low compared to the contaminated site, which means that these chicks were not symmetric at all in this area. And I used all this data to make a new synthetic variable based on all the parameters she measured. And we demonstrated that there is a correlation between the progeny abnormality, that is a new synthetic variable, a significant relationship with the chick physiological status. So with the idea to make the relationship between biomarkers and disturbances at higher level of biological level. There is a significant relationship. So that's something I'm going to write this year. So what could be the potential cause of the chick morphological abnormalities? It could be due to a direct exposure to micropollutants, because seabirds are top predator and are prone to bioaccumulate. And so if they are exposed to micropollutants during their early life stages, it could lead to abnormal development. But it could be also linked to transgenerational effect. Maternal effect and paternal effect. And actually, I'm very interested in this too. Like the maternal effect will be that if the female were feeding during the reproduction on contaminated area, they may have accumulated lipophilic contaminants in lipids. And when they produce the eggs, these lipids, these lipophilic micropollutants could have been absorbed to the vitellus. The vitellus is a yellow eggs part of the egg, and that is used by the embryo to develop. And so if this vitellus is loaded with micropollutant, at some point, if there are genotoxicants, they could be remobilized and induce genotoxicity. And at these early life stages, deleterious mutation could have long-term effects. And it's the same with the paternal effect. During my PhD, I worked on biomarkers, but most focused on genotoxicity, and males are prone to accumulate if males are exposed to genotoxicant, they could accumulate deleterious mutation in germ cells, in spermatozoa, and this deleterious mutation could be transmitted to the progeny, leading to abnormal development. And these three parts are definitely projects. So I've done a few projects about that, so we could talk about that later. But and it's something I would like to develop more in the future to understand these long-term transgenerational effects. So thank you for your attention. And if you have any question, it would be a pleasure to try to reply.
Are you going to apply your ugly cute index to fish? It's cool.
That's possible. Yeah.
It's interesting stuff. I like that. Yeah.
Thank you, Keller.
Yeah.
During my PhD, we exposed fish to genotoxicants and crossed contamination and made in vitro fertilization to assess what is the parental effects, maternal effects, and paternal effects. And actually, when males are exposed to contaminants, as spermatozoa do not have any defense in terms of DNA repair systems, we could carry the deleterious mutations that are transmitted, and some of the exposed parents, when I checked the morphological abnormalities of the progeny was even more ugly than the chick in the study. Yeah, they suffer from some skeletal deformities, and some of them couldn't swim, like they were swimming. Yeah, it was like basically the kind of individuals that are selected very early in the environment. Well, I don't know if you have any question.
Any other questions, Barry?
You mentioned in the first part of your talk that there were 3 species that were locally extinct or totally extinct in Lake Geneva, was it?
Yeah, totally extinct.
Okay, are they just only known from Lake Geneva?
They were in 2 lakes, except Coregonus ferra, that was endemic from the Geneva Lake, and it's extinct.
Yeah. Is there any program in Europe to reintroduce species from aquarium stock or captive fish in zoos? Or
what they did is that they reintroduced a fish species from the lake of Neuchâtel, from the Neuchâtel Lake in Geneva, and even if it's not so the extinct species is Coregonus ferra, that was called La Ferra by fishermen. And what they did is that they reintroduced a new species and they called it Ferra as well. So even if the true Ferra is extinct, as it happened like decades ago, with time, people forget that it's not the same Ferra, but it's a new species. It's a species from elsewhere. Yeah.
And I'm also curious to know that microplastics, there's been a lot of discussion about them in the marine environment. How important are they in freshwater environments?
In where? In
microplastics in freshwater environments.
There are plenty of studies about that, and when actually the system, the apparatus we have designed could be used to assess the impact of microplastic, because it could resuspend any particles. And in freshwater, it's also a main issue. And when I left 2 years ago, almost 2 years ago, there was a postdoctoral project on the impact of fine particles of tiles. Because the Geneva Lake is around the Geneva Lake, there is a lot of traffic, and so they were investigating the impact of this microparticle on fish species. So it's definitely a very relevant topic in freshwater as much as it is in marine sciences.
I was wondering about. Thank you for a fascinating seminar. It sounds like you're using quite a lot of technical expertise to do this monitoring, and I was wondering if you're aware of programs that are looking at these things from a citizen science point of view and how accessible it is to do this kind of research on a citizen science approach.
Oh, I don't know about programs that include citizen science, but I know that it's something we should more develop for sure, because for now the program is based on electrofishing programs that are standardized, and you need to have a license to do this project and to do the electrofishing. But it's definitely, it would be interesting to develop more programs based on citizens science, and especially I will use these results to communicate with the community of recreational fishermen to develop more projects with them, especially definitely. And we should definitely do that, because it's a good way to get information and the best way to spread the education. So
Great.
Any questions online? There's a few people online. Silence. Thanks, Raphael.
thank you,
Everyone
Taylor.
put their hands together.